We will continue with the data blitz presentations. They are split into two groups. Group A includes 3 talks, which are related to COVID-19, while Group B contains 5 talks. Sorry for the mask. 5 talks which are related to technology. Let me also say here that all data blitz presentations have been videotaped. So this means that the data blitz session will be separated into 4 parts. In the first part, we will attend all 3 Group A talks and then accept questions regarding Group A talks, and then move to part B of Data Blitz talks, followed by another Q&A session. So we will start with the presentation of Talk A talks. Are we ready?
Good afternoon, ladies and gentlemen. My name is Flora Nicolaou, and I'm currently clinical psychology PhD student at the University of Cyprus. The project I will present you today was done during the lockdown period under the supervision of Professor Fovi Constantinidou and has the title Epidemiological Insight of Psychological Impact and Compliance to the Precautionary Measures among the Elderly Population during the COVID-19 pandemic. Thus, the key questions of the present study were how this population had been psychologically affected due to the pandemic, what are the rates of compliance to the precautionary measures by this population, and what is the psychosocial resilience manifested by these individuals. So, our main objectives were to determine the prevalence of psychological symptoms in elderly individuals during the lockdown period, to investigate the compliance to recommended precautionary measures, and to investigate the resilience and coping skills of the elderly. That the 56.1% reported high levels of resilience, 35% medium, and 8.9% reported low levels of resilience and coping skills. An interesting finding of this study was the negative correlation we found between resilience with both anxiety and depression scales. Last but not least, regarding the precautionary measures, gender difference was detected, showing that men had lower compliance to precautionary measures than women. Age differences indicated that the older age group, namely 75 and above, reported lower levels of compliance with precautionary measures than the younger age groups, namely 35 to 74. Also, correlation analysis revealed that there was a small but statistically significant correlation with telephone mini mental status examination and the precautionary measures. precautionary measures. Lessons learned from this acute crisis not only inform our understanding of this population in general, but also prompt public authorities in order to pay more attention to these individuals, providing more interventions for mental health and guidance for the implementation of precautionary measures.
Hello everyone, I am Evangelia Tedolouri. My presentation today relates to a study conducted by the Department of Psychology and Center for Applied Neuroscience on the possible impact of COVID-19 confinement measures on children and adolescents. Recently, we were all asked to cope with a major challenge which involved our and our close family members' protection from the pandemic COVID-19. As a result, in Cyprus, we were asked to comply with the government's strict confinement measure for 70 days. Children and adolescents were equally called upon to cope with confinement measures. The sudden closure of the school, the social distancing, and the reduction of physical contact or the discontinuation of participation in joint activities with other people were some of the most important challenges to which we all had to respond and adapt almost instantaneously. It is known that this kind of physical isolation is likely to lead to the images of high-risk mood-related mental disorders, such as anxiety, depression, and thought disorders, as well as symptoms of post-traumatic stress disorder associated with confusion and anger. Thus, the present study was conducted to help mental health professionals, educators, and parents alike to understand how children and adolescents experience confinement or lockdown measures as a consequence of the COVID-19 pandemic. Regarding the study's methodological aspects, we first sought approval by the National Bioethics Committee and the Cyprus Ministry of Education, Culture, Sport and Youth. Second, the data collection was carried out for 3 weeks, when both Cyprus and Greece started to gradually lift their containment measures with a view to entering the recovery phase. Participants were asked to respond to a bespoke 15-minute online questionnaire through the Red Cat platform to record the experience during the lockdown period. The survey also includes some exploratory questions about their future emotional state, focusing on post-traumatic stress disorder. Finally, the study was spread to the public via several social media channels. The final participant cohort comprised 1,061 children and adolescents aged 5 to 18 years from Cyprus and Greece. And now we are going to results. Demographics. Participants were equally distributed from both genders and countries. The majority, 91%, come from 3 or 4 member families, and the rest, nearly 9%, from extended families with more than 4 members. Participants had no diagnosis for COVID-19 infection, and the majority, 98%, responded that the same was true for their close relatives and friends. Basic and general psychological needs. The participants' basic needs such as sleep, feeding, and the sense of home security were not negatively affected as stated by the vast majority, above 80%. Also, the majority of the participants expressed their concern about the possibility of a close relative getting sick, which was comparatively higher when thinking about themselves getting sick. The need for social contacts was evident in both children's and adolescents' responses, as spending time with relatives and friends was something that they much missed. The two age groups differed significantly in the use of alternative ways of communication, for example via computer, mobile phone, or tablet, with adolescents spending considerably more time in front of a screen per day. In contrast, children communicate their emotions, such as joy or sadness, with family members more directly than adolescents. During the quarantine, the main entertainment activities for children and adolescents center around electronic games, gymnastics, for example cycling, walking, pet walking, gardening, such as planting, watering, and cleaning, and indoor activities. A smaller percentage of children play board games at home. School. The majority of the participants, slightly above 80%, attended distance learning courses. Nevertheless, they did not express satisfaction from this alternative educational experience, particularly with the degree of their concentration during distance learning. Regarding return to school, most children, over 63%, want to return as soon as possible, whereas only a small percent of adolescents did so. The need for socialization and contact with relatives was apparent in both age groups, and this is what they would immediately do as soon as the confinement measures were lifted. Future emotional state. There was successful management of the psychological state and related emotions. The pandemic did not cause any significant fear, grief, hopelessness, or anxiety, with adolescents to be more introverted and share the experience predominantly with friends. In conclusion, both children and adolescents were able to cope with the challenges the confinement measures brought with them, showing no distressing PTSD symptoms. Factors such as individual characteristics, family conditions, and the support they received from the environment seem to have helped to self-regulate emotions and deal with the situation with high degree of adaptability. Children and adolescents showed that we have the psychological tools to manage stress successfully, adapt to the pandemic's uncertainties, and stay mentally healthy. Thank you for your attention.
I'm Marios Anastasiou, and I will present to you the report with title Public Interest in COVID-19 in Cyprus, Evidence from Google Search Trends, a report that I have worked on with Katerina Pandavou, Aneza Yalourou, Stefano Bonova, and Giorgio Nikolopoulou. The purpose of our research was to examine Cyprus public's online interest for coronavirus disease, overall in Cyprus, as well as by city of Cyprus, Nicosia, Limassol, Larnaca, and Paphos. Also, in the scope of our research was the examination of some of the top coronavirus symptoms, namely fever, cough, sore throat, shortness of breath, and myalgia. Let's now explore the methodology followed. Firstly, the data analyzed were downloaded from Google Trends, a website by Google that analyzes the popularity of top search queries in Google Search across various regions and languages. Then, a preliminary popularity assessment was implemented between the terms you can see in the oval at the left-hand side. From that assessment, the most popular query was found to be the word coronavirus. That term had the highest popularity metric. Note that the popularity metric that shows how popular a term is called interest score, and it is a normalized measure to the time and location of a query, ranging between 0 and 100. The examination period was the first months of the pandemic, specifically from the 19th of January to the 18th of April. In this slide, all the results are illustrated, but let me present to you the key results. As you can see on the top left graph, the top public's online interest in coronavirus was between the 10th and 15th of March, during a period where the first case in Cyprus occurred and the borders of the island closed. There were two lower peaks. One at the 31st of January, where the World Health Organization declared global public health emergency and Italy declared state of emergency. Another peak occurred at the 26th of February, where 10 Italian cities were set on a lockdown and Greece had their first case. Also, at the top right graph, public's highest interest was found in Paphos on the 13th of March, with an interest score of 100. Note that 2 days previously, on the 11th of March, a taxi driver was diagnosed with COVID-19 in the same city. Moving to the symptoms exploration, from the middle graph at the bottom, you can observe that the most popular symptoms were fever, with a weekly average interest score of 57 between the 15th and 21st of March, and cough, with an interest score of 49 during the same period. So, based on our research, the conclusions are firstly that Google Trends can be used to identify periods of high public interest in COVID-19, and symptoms with the highest interest were cough and fever. Finally, high interest in COVID-19 seems to be related to announcements of governments or health authorities. Therefore, authorities should constantly monitor and evaluate public's interest to respond to epidemics. Google Trends is a tool that could contribute in order to provide accurate and reliable information to the public on time. Thank you so much for your attention.
Okay, hi everyone. I hope you're all well and you can hear me. We're going to continue with another 4 presentations for the data blitz. The first one is by Evgenia Kouki on multilingual eye movements corpus data from Greek or from Greece.
Hello everyone, my name is Evgenia Kouki and I will present you data from Greek deriving from the multilingual eye movement corpus. This project is conducted in collaboration with McMaster University and Haskins Laboratories under the coordination of Professor Papadopoulos from the University of Cyprus. It has been shown that proficient reading depends highly on integrating eye movement patterns and linguistic components within a limited time window, and therefore eye movement patterns are a strong predictor of reading fluency. That being said, increased effort in text processing has been reflected in previous research as differentiations in eye movement patterns, such as longer fixations. Therefore, the MECO project aims to explore how different patterns of eye movement inform the hypothesis about the role of orthographic complexity in reading, and which of the eye movement variables best account for variance in text reading fluency. In Figure 1, you can see the setting of an experiment with the use of eye tracker, where the camera identifies and records the pupil's movements across the screen. For the purpose of the study, we recruited 45 adult readers who read silently 12 texts taken from Wikipedia entries, edited for length and complexity. Text length ranged between 135 to 201 words, and 4 yes or no questions followed on a separate screen to ensure that the individuals had read the texts carefully. Monocular tracking with a sampling rate at 1000 Hz from the right eye was recorded using iLink 1000, and viewing of the display was binocular. Data were analyzed with the Popeye software, and verbal and non-verbal ability were also assessed with the CFT20 and Greek vocabulary test, respectively. In Figure 2, you can see an example of eye movement recordings. The blue color represents fixation sequences across text during reading and illustrates how participants' gaze fixated on various parts of the text. A hierarchical regression analysis was conducted, and results are summarized in Table 1. General ability accounted for only 3% of the variance in text reading fluency, whereas gaze duration and number of fixations added a further 87%. Standardized beta coefficients for gaze duration and number of fixations demonstrated that there was a significant negative relationship between eye movement predictors and reading fluency, and therefore indicated that the longer fixations and increased gaze duration were observed in reading more demanding word strings. In this work, eye movement patterns were investigated in text that resembled everyday reading in terms of comprehension demands, and our findings coincide with previous evidence showing that gaze duration and number of fixations contribute significantly to text reading fluency. Concerning future directions, we also aim to investigate whether the same eye movements are potential predictors of reading fluency in readers' second language, which is most often English, and also examining word frequency and word length could further provide information on the eye movement components best predicting reading fluency. To conclude, eye movement research in reading can provide detailed information for the development of reading prediction models that will withstand cross-linguistic comparisons. Thank you for your attention.
The effects of transcranial alternating current stimulation on movement execution in Parkinson's disease patients. This constitutes an ongoing study, as the recruitment of healthy controls has been rather challenging given the extraordinary circumstances. The activity of the primary motor cortex has a different electroencephalographic trace during movement and during rest in healthy subjects. Beta oscillations are considered antikinetic as they become prominent at rest or tonic contraction. Gamma oscillations, on the other hand, appear with voluntary movement and are thus deemed prokinetic. Transcranial alternating current stimulation is a non-invasive brain stimulation technique which has been demonstrated to modulate cortical rhythms in healthy subjects. TACS, as you can see in this illustration, involves the application of an alternating current by surface electrodes attached on the subject's scalp. Parkinson's disease patients demonstrate pathologically enhanced beta oscillations in cortical and subcortical regions, which correlate clinically with a degree of bradykinesia. Bradykinesia is a cardinal motor feature of Parkinson's disease and refers to the slowness of movement and the progressive decrement of velocity and amplitude with movement repetition. The present study aims at investigating the effects of beta and gamma attacks applied on the primary motor cortex on bradykinesia in Parkinson's disease patients and the underlying neurophysiological mechanisms. The study enrolled 15 Parkinson's disease patients, each participating twice, once on and once off levodopa treatment. The first part of the study involved a kinematic recording of finger tapping during tags. TAGS was delivered as a beta, gamma, or sham stimulation in a randomized double-blind fashion. An optokinetic system was used to record the movement parameters of finger tapping during TAGS. The second part involved transcranial magnetic stimulation during TAGS. TAGS was again delivered as a beta, gamma, and sham stimulation in a randomized double-blind fashion. 3 TMS protocols were used to obtain motor-evoked potentials. Single pulses were used to study the corticospinal excitability. Short interval intracortical inhibition, or SICI, was used as a surrogate for GABAergic circuits, and short interval afferent inhibition, or SI, as a surrogate for cholinergic circuits. Starting with the movement parameters obtained during the finger tapping task, our results indicate a significantly increased amplitude during gamma tags and a significantly decreased velocity during beta tags. The frequency-dependent effects of tags were independent of the on or off levodopa state. Moving on to the neurophysiologic parameters obtained using TMS, our results indicate that motor-evoked potentials obtained with CG were significantly higher during beta and gamma tags compared to sham, and that motor-evoked potentials obtained with Psi were significantly higher during beta tags compared to sham. Again, the frequency-dependent effects of tags were independent of the on or off levodopa state. Moreover, there were 2 correlations between movement and neurophysiologic parameters. First, during gamma tags in the off session, CG MEPs were directly proportional to the amplitude. And second, during beta tags in the off session, CG MEPs were directly proportional to the velocity. In conclusion, our results suggest that bradykinesia was modified by tags in a frequency-dependent fashion, and this constitutes a novelty in literature. We theorize that the increased amplitude during gamma reflects a prokinetic entrainment of the cortical activity in the primary motor cortex, while the decreased velocity during beta tags reflects a resonance with the antikinetic native oscillations. Finally, the proportional changes in neurophysiologic parameters may provide some further insight. Specifically, we speculate that the suppression of intracortical GABAergic circuits during gamma tags may be responsible for the increased amplitude, while during beta tags it may reflect a compensatory change to counter the enhanced beta activity. Thank you for your attention.
And today I'm going to present you the project entitled Intelligent Patient Assessment and Monitoring System, Developing an Emotion Recognition Algorithm for Vocal Cues. Let's start with the objectives. The aim of this project is to develop an intelligent patient assessment and monitoring system consisting of having different sensor networks like a USB microphone, web cameras and wristbands, also having smart software algorithms that will monitor and assess individuals with TBI in the rehabilitation facility. Most importantly, this study is part of the IPAMS project, and the purpose is the development of a sophisticated online algorithm that will detect and classify vocal signals into different emotions. For this project, we recruited 24 neurotypical participants aged between 18 to 51 years. We used the 3.7.6 version of Python And we also used the Ryerson audiovisual database of emotional speech and song dataset in order to develop our algorithm. The code of speech expressions included happy, sad, angry, fearful, disgust, surprised, and neutral. The gender differentiation was inserted to detect the different emotions between men and women. And most importantly, the output is a real-time speech emotion recognition. According to the results, the speech recognition algorithm developed and implemented was shown to effectively detect and classify all targeted emotions in men and women. It is noteworthy to mention that this speech recognition technique can be applied in real-time emotion detection. In other words, we have the input, which is the participant speech using a USB microphone, then the real-time speech detection, and at the end, the predicted output. These findings claim that real-time voice processing algorithms could contribute to the understanding of the emotional state of individuals more efficiently prior to the presence of any disruptive behavior. Indeed, this software algorithm can be further developed to identify a potential behavior alert through a personalized profile and to notify the rehabilitation setting and the individual with TBI with real-time feedback. The expected outcomes can be applied in areas beyond health where real-time monitoring and assessment is required, such as educational programs. Thank you for your attention.
And the final talk will be by Maria Penido on association of AB plaques and connexins in the spinal cord, again in Alzheimer's mouse models.
Hi everyone, my name is Maria Pechlevanidou. I'm a PhD candidate in neuroscience at the Cyprus School of Molecular Medicine, and today I'm going to give a short presentation of my PhD experiments with the title Glial Connexins Expression in the Spinal Cord of 5xFAD Alzheimer's Model. To begin with, the 5xFAD mouse model is a model that represents the familial Alzheimer's disease, the early onset, and the aim of the project is to explore the amyloid pathology along with alterations in the expression of glial cells in the spinal cord of that model. And glial cells, we refer to the oligodendrocytes, astrocytes, and microglia. The objective is to assess motor learning and coordination in 5xFAD and wild-type mice, to examine the concentration of the extracellular beta-amyloid plaques in the spinal cord, and measure the degree of astrogliosis, neuroinflammation, neuronal loss, and oligodendrocyte loss in both gray and white matter of the spinal cord. And the research hypothesis is whether there is an association between amyloid pathology, astrogliosis, white matter changes, and motor deficits in 5xFAD mice. Because people with Alzheimer's disease show also symptoms of motor decline. The methodology involves experiments in 3 and 12-month-old 5xFAD and wild-type mice, where we perform motor performance tests like rotarod, footprint, foot slip, and the hind limb response to assess the learning and the gait of the mice. Also, fluorescence immunohistochemistry experiments in the following cervical and lumbar spinal cord sections, as you can see, for the following markers that represent, for example, that one, the beta-amyloid plaques, the GFAP and IBA for the astrogliosis, thus the neuroinflammation, the CC1 and the OLIC2 is for the precursor and mature oligodendrocytes, and UN is for the neurons. Here is a few of the results that we obtain. In the picture here, you can see the footprints of both 3-month and 12-month-old 5xFAD and wild-type mice, and we assess the stride length and the width, and you can see that they have abnormal gait. In that picture that you see those two mice is the hind limb response. You grasp the mouse from the tail and they perform the response where they grab both the front and the hind paws very close to their body. And the wild type mice, they just spread their legs, as you see over there. The immunofluorescence pictures represent the extracellular beta-amyloid plaques that are abundant in 12-month-old mice, as you can see from the green dots. And the pictures here is the GFAP and the IBA, where we see severe astrogliosis and neuroinflammation in the older mice groups. In those two graphs, we can see the significant neuronal loss that we obtain in the 12-month-old 5xFAD mice. Also, that picture over there, we can see the 3 different oligodendrocyte populations that we counted in the spinal cord, and there is significant loss of oligodendrocytes only in the white matter of the spinal cord. And in general, the 12-month-old 5xFAD mice have impaired motor performance in all the tests. To conclude, we have severe astrogliosis, significant neuronal loss and oligodendrocyte loss of the mature oligodendrocytes in the white matter of the spinal cord of 12-month-old 5xFAD mouse. Those pathological changes probably contributes to the overall Alzheimer's pathology and neurodegeneration, and probably to the related motor decline. So future experiments will be conducted to investigate further the molecular mechanism and the contribution of the glial cells through the gap junction proteins to the related myelin and axonal damages, and to the motor deficits that we observed. That's it. Thank you very much for your time.
Okay, so I think we're done with the data blitz presentations, and we do have some time for the presenters to respond to questions. I assume they're all online at this time. So does anyone want to start with a question to any of the presenters of the A and B presentations? Can I ask a question then, if no one else wants to ask something? So my question goes for Evgenia. Evgenia, are you there?
Yes, I am.
Okay, so so great presentation. My question is about your results. So you present the results that are based on the Greek text. Do you find interesting things, but do these things differ from the results that are commonly found in reading studies in other languages, and especially English, where there is more research on English than any other language, I assume?
Actually, this is what we are going to study further, because we have some data on how the same readers processed texts in English, and so I don't have any preliminary results to discuss with you at the moment. However, what was found was that in the existing literature, what was found was that orthographic complexity played an important role. And as far as the Greek language is concerned, we found that more fixations were accumulated on words that were were longer or less frequent, and this is something that we also aim to study further in an area of interest analysis. So at the moment, these are the data we have.
Okay. All right. Thank you, Evgenia. Josh Spanoudis is asking a question on the chat, so maybe I can read it. He says, I have a question related to the second talk of Part A talks, which is the one by the possible impact of COVID on confinement measures on children and adolescents. So the researchers found no effect of lockdown on dependent variables of the research design. This finding is contrary to the data of other investigations. Why? How you explain the difference?
Hello, is for our presentation about COVID-19.
Yes, yeah.
Okay. I think that we found this difference because of the ages of the participants, because we have only children and adolescents, and I think it's because of the age. Because we see in other researchers like in the same topic that the people in older ages have more stress. So the COVID make them more stressful and more anxiety, more other things like this. So I think that the children were more not free, but they understand all the fact in other way, I think.
Okay, thank you, Evangelia. Another question to Flora.
Yes.
Yeah, Flora. So, can you read the question? Can you read the question that?
Yes. Yes, of course.
Okay. Because there's 3 questions, and I don't want to repeat them.
Yes. Okay. So, as far as concerned the question of Dr. Fofi, we found that there is a correlation between cognitive status that was measured by telephone mini-mental status examination and precautionary measures. Therefore, cognition does matter for the compliance to the precautionary measures. I'm not sure if I'm answering the question of Dr. Fofi, I think.
Okay. Well, Fofi is listening, so if she wants to
Yes.
jump in and clarify, that would be great.
Okay, can I say something?
Yes.
Thank you. So I guess the question, Flora, with the study with the older adults has to do with the fact that we found different effects for older adults versus younger adults. And so the question is if there is a link between cognitive performance and the use of precautionary measures.
Yes. As I said, the cognition, which was measured by mini-mental, shows that there is a relation with precautionary measures. Older adults showed that as the higher the age was, specifically if where participants at the group of 74 and above showed less compliance to the precautionary measures than the younger age groups, namely the 36 to 74 adults.
Okay, thank you, Flora. There's another question on the chat about the children presentation. I assume is again Evangelia's study. Can you please provide detail on the methodology you used for the data collection? So Evangelia, if you're still around, can you provide some information about how you did complete the data collection for your study?
Yes, of course. It was an online questionnaire through the Red Cap platform. Is that it was online? Yes, and we send it to we sent it to the people all over the Cyprus and Greece, and whoever wants to answer, it was free to answer or do not answer. And if someone answered the questionnaire and don't want to and change the mind through the time, he can to delete it.
Okay. Thank you, Evangelia. Any other questions? Can I ask you a question then for the final presentation? This is the one that I had a bit of a hard time following, given that I don't have a background on the topic. So Maria Peklibanidou, you're still around. Could you give me like a very general audience description of what the main result is? That is, what's the implication, in other words, for your findings for understanding Alzheimer's disease?
Okay, hi. I study the pathology of Alzheimer's in the spinal cord. So in the spinal cord, there are only motor neurons, let's say, and the spinal cord is responsible for the motor performances. So the implications that we observed through the motor performances test is that our mouse model has motor decline with age. And that is also found in real patients with Alzheimer's disease, that there are cases that they have motor decline even at preclinical stages, before the cognitive impairment. And there is a paper, actually a few papers, that they show that the motor deficits appear before the dementia. So we wanted to explore at the model, of course, level, if there is a correlation with the glial cells, and particular the gap junction. There are proteins on the cell membranes that are responsible for the communication. And the overall, we found that there are some pathological features like neuronal loss, and the glial cells, they get activated and reactive, and there is severe inflammation. And that's the initial finding, together with the bad and low motor performance of the older mice. So we will continue to build a stronger association and find why those mice have motor decline, and why that happens also in humans.
Okay, thank you very much, Maria. Great work. So unless anyone else wants to ask a question, I guess we could stop here. We're already 6-7 minutes past the time, and so unless anyone wants to ask something, last chance, we can go to the final remarks and the electronic poster presentation. So back to you, Sarah.
I guess just a few words before we adjourn and move on to the poster presentation. I want to thank all of you on behalf of our colleagues at the Center for Applied Neuroscience. Also to thank the live audience. And hopefully next year we'll have a chance to have a true live event, in-person event. I'm sure we'll maintain the online presence because we've had a lot of people attending from all over the world and through our Facebook live streaming. But please, those of you who have registered for the poster sessions, stay for those discussions. And while we are engaging in physical distancing, hopefully we're not socially isolated, but we're all connected. Thank you to all the panelists, Data Blitz, and poster presenters.